Thank you, Kathy, and thank you very much for inviting me to talk at this conference. It's a great privilege to be here. I had some discussions with the organisers about what I should talk about, and the title, Organisation of New Drugs in Childhood Cancer, obviously captures quite a lot of things, and I could happily talk for a couple of hours on that, but I won't, I promise. Honestly, Kathy. What I thought I'd do is actually talk a little bit about the framework in which we actually develop new drugs. And I think the key questions that always get asked are, why haven't we got more new drugs for children, and why does it take so long? So what I hope over the next just under 15 minutes, I can just tell you a little bit about what it takes, what the steps are to develop a new drug, not from the laboratory end, but from the clinical end, and hope that starts to answer some of the questions as to why it is so long and so complicated. So I'm going to divide this. Principally, I'm going to focus on the pathway. I'm going to touch on a couple of slides about the law that regulates it, and then I'm going to just explain some of the networks in which we work. So the pathway. You'd think it would be straightforward. We know extraordinary amount about the tumour biology, particularly in childhood cancer, and that's increasing week on week. We have lots of ideas. We have lots of people interested in drug development in the childhood cancer arena, and as a director of the trials unit, we get probably a thought a week from people about what we could potentially do. And we have access, and increasing access, to the new adult cancer drug arena. So the elements are all there, and you think, well, we should from that have a pipeline of new childhood cancer drugs that was fairly steady. But the problem is, it is a long and tortuous road, and it is necessarily long and tortuous, I'm afraid. But what I hope I can show you is how we're beginning to start to shortcut that a bit, or at least find ways to get down this road a little bit quicker. And whatever we do, we have to remember to balance the toxic effects of drugs against the tumour effects. And obviously, we want the balance to be towards the benefit in tumour effects. But every stage of clinical drug development, we have to make sure that the balance is in the right direction. And that is part of why it all takes so long. So this is the sequence of drug trials from the moment we get a new drug out of the laboratory, and for us in paediatric cancer, out of the adult cancer arena. We have to go through a stage of phase 1, phase 2, phase 3, prior to marketing authorization, and then phase 4. And I'm often asked, what does that mean? So I've translated that into English. And what this actually means is the first stage, phase 1, is a safety and dose level, so trying to identify what the drug actually does to a patient, then work out if it might work, then work out if it's better than what we already have, before we even consider marketing authorization. And phase 4 studies we don't do very often in paediatrics, but this is collecting data on the outside of normal clinical trial use of a drug. So what I want to do is just explain how each of these studies are designed, and it gives you an idea of the kind of timescale it takes. So we get a new chemotherapy drug. For children, it is almost never first in man. And I say almost never. There are some now examples of drugs that are being designed specifically for childhood cancers. But mostly they come from having been tested in adults first. So we know something about them. But we still need to know what's the best dose and what's the best dose to use in children. And by best, we mean the safest dose and the most likely to work. And in the old days, that just meant the dose that had the least side effects and the one that makes the tumour shrink. Whoops. But nowadays, most likely to work in one of these early studies might be just demonstrating it hits the target. The drug might be targeting a particular chemical in a tumour cell, and we want to see, does it actually make that target turn off? And that might be all we're testing in that early phase study. And there is a classic design for this, which is called a 3+3 design. And although we've moved to slightly more innovative designs, the principle is pretty much the same. So if you imagine each of these squares is a patient, we take the first dose level, which in paediatric studies is usually very close to the dose that's been decided to be safe in an adult trial. We put in usually for a small number of patients, 3. We look to see if there's any side effects in those patients. If there is a significant side effect, we expand that. If we're still seeing significant side effects, then we wouldn't go any higher. But if we're not, we expand to the next dose level. And we continue this until we hit the dose that is safe and hitting the target, whether that be shrinking the tumour or just hitting a biological target. As I said, we are getting more innovative designs, but the principle is the same. But the innovative designs are trying to get through this pathway quicker. And when we get to the dose that we think is safe, we often extend the study a bit to learn a little bit more about how the drug's working in a slightly larger number of patients. And that's shifting towards the next stage of a trial. And the next stage is the phase 2, what I call a signal of activity study. These trials, usually about 20, sometimes up to 60 patients, in a single disease. So these studies are done often in a multitude of diseases, like all patients with a lumpy disease or all patients with leukemia. These ones tend to focus on a single disease, Ewing's, neuroblastoma, or type of brain tumour. And what we're asking is, can we hit a certain target number of responses? And again, remember, we're balancing toxicity against tumour effect. Do you think, surely we want a drug that goes green on every patient that goes in the study? But that's not realistic. Most of the patients that go into these studies have had lots of other treatments. They failed those other treatments. And we're looking to see if we can find a drug that in those resistant diseases shows an effect, but there's no guarantee. And the percentage of green lights that we get out of the total population is determined by an expectation on that drug. And that expectation is, again, a balance of tumour effect against toxicity. If we hit the target, and the target might surprise many of you to know, could be as low as 20%. A 20% response rate is enough to say that looks like an interesting drug. But that is by no means enough to say this drug now should be rolled out to every child with that cancer. Because, one, we don't know enough about it, and two, we really want to know is, can this drug be taken forward into children who've not received all conventional chemotherapy, but actually could potentially have a better outcome if we use the safer and more effective new drug. So how do we do that? Sorry, before we go on to that, I'm just going to give you an example of a phase 2 study. This is a drug that has been around for a while. It's not a standard chemotherapy drug. It's an antibody. It's targeting blood vessel development and is already licensed in adults. There's been early safety studies done in children, and there's a lot of biology to suggest that it might work in childhood cancers, and particularly in neuroblastoma. So we're running a study across Europe called the Beacon trial, which is a study in children with neuroblastoma that have failed current treatments. And rather than just doing one phase 2 study, we're doing parallel phase 2 studies, testing drugs that are not part of frontline treatment at the moment and combining it with the new agent. And the question we're asking is, which of these drug combinations should we take forward? Can they be safely combined, and which is going to be the most effective to take to frontline studies ultimately? Now, that's a study of 4 arms with 3 different drugs. But while the study's been open, another study's been published suggesting that topotecan in combination with temozolomide is an interesting combination. So the study's been amended, and it now has 6 arms, asking some more questions. And this study will roll on, so every time we get a new agent or a new combination that's promising in neuroblastoma, we can amend the trial and look for new drugs. So what we hope is we have an efficient way of taking new combinations forward. But when we get that, which is the best, we need to say, is it better than what we're doing today? Because all children with cancer are on standard treatment protocols, and can we improve them? Because it's all very well doing new agent studies, but we want to make them the gold standard of the future. And we do that in these so-called phase 3 studies, where we compare current best treatment against the current best, modified in some way by a new treatment, either replacing a current drug with a new treatment or adding it to conventional treatment. And this is done in a randomized way, comparing 2 populations of patients between these 2 treatments. And the only reason we can do this is because the difference has to be at the beginning that we believe is an equipoise. And by that I mean that we don't know which is the better, and the evidence suggests that there isn't one that's going to be obviously outstandingly better than the other. Otherwise it wouldn't be ethical to do this. But it's important to do this slowly and incrementally so that we know whether the new drug adds benefit to what we've already done, or whether it doesn't make any difference and is just adding side effects. This is an example of a phase 3 study that we're going to open this year. And if I take you back, this is the R, the randomization. This study, we're asking 3 randomized questions within the trial. So we don't do it just in one question per trial, which takes 10 years to run. We do multiple questions. And this trial is adding a new agent right up front, frontline myeloid leukemia. This is an unlicensed drug that's been through its phase 1 and phase 2 studies and some phase 3 studies. And we're now asking the question, can we add it to improve the treatment in frontline? Ultimately, if this trial is effective and shows that this drug, adding it, is better for children with myeloid leukemia than current treatment, this will become the new gold standard. So that's the pathway. It's regulated by 2 laws, Clinical Trial Directive, which is going to be called the Clinical Trial Regulation soon, and that law protects patients in trials and protects the integrity of the trial data. And the other law is the Paediatric Regulation, and the Paediatric Regulation was designed to promote development of new drugs for children, all children, not just for cancer. Both are highly bureaucratic, but actually both help us. Paediatric regulation has improved this pathway. It used to be that a drug would have to go right the way to being marketed in adults before we could get it for children. The paediatric regulation creates a shortcut. So an earlier phase in the development in adults, the companies have to develop what's called a paediatric investigation plan. I'm not going to go into that in detail. It'll be a talk in itself. But just to say that by having this new law, which came into force in 2007, there are now increasing drugs coming into paediatric development. The law is not ideal. There are limitations, but it is increasing the access to drugs. So what networks do we have to deliver these trials? Well, in the UK, many of you know, we have 21 centres in the country that are specialists for treating childhood cancer. We have one trials unit, which is designated as the Children's Cancer Trials Unit. And within that trials unit, we have a dedicated team, the Childhood Cancer Trials Team. It's unique in the country, and it coordinates the trials. And we have a specific part of that team that are working on the new drug development trials for childhood cancer. This is our portfolio at the moment. These are open and recruiting studies. The ones in red are ones that involve a drug development element. These are the ones that are all funded, so will open in the next year. And these are the ones sitting on the desk in development. So you can see the development of new drug trials for cancer, for childhood cancer, is very healthy. That's just the academic trials being run from our trials unit. If you look at this in terms of the pharma trials, this is data from the middle of last year, there is a huge number of pharma trials adding to that portfolio as well, early phase trials developing drugs for childhood cancer. So it is very healthy. Not all 21 centres are able to deliver the early drugs trials, but we have a good network of now, I was going to say 10, I think it's 11 now, centres across the UK that are part of what we call our Experimental Medicine Centres network that are delivering those early phase studies. But we can't work alone. We work within Europe, and with this particular organisation, academic European organisation called Innovative Therapies for Childhood Cancer, and that provides a network, including our centres, of 47 centres across Europe, and in our definition, Israel's in Europe. So this is our network to deliver those childhood cancer studies. We, in the ITCC, deliver the early phase studies, but remember I said it's about practice-changing phase 3 studies. They're delivered through the networks for specific diseases across Europe, including the neuroblastoma network, the blood cancer network, the sarcoma network, and the brain tumour network, and there are many more. So we have a pipeline going from early phase right through to phase 3. And coming back to the Beacon trial, you remember the Beacon trial with the 6 arms? This study is not just a UK study, it's a European study with funding from Imagine for Margot, and we'll hear from Patricia soon, to help us deliver this in Europe. And you can see, by bringing all those elements together, we are now able to sponsor trials that run within multiple countries and in multiple sites, and this allows us to accelerate that delivery. And we're repeating this sort of network, or this sort of trial, for other diseases. We've got one called Recur for Ewing sarcoma, and we hope to be able to run similar trials for other diseases within the ITCC network. So none of this is done alone. This is from our last annual general meeting. You'll see there's a huge number of people investing time and effort in the ITCC, and these are the staff at the trials unit and the Children's Cancer Trials team. Thank you very much.